Hi everyone, in this experiment we'll be testing for starch or complex carbohydrate as some people will call it. Carbohydrates in general are also known as energy giving food, and the reason why they are called energy giving food because they provide energy for the body to perform useful work, and is also an essential component in a balanced diet. In this experiment you will need 6 food samples or more. You also need a negative control just to increase the confidence in the result of your experiment, and as you know, the positive control will be what you're actually testing for, and in this experiment we're testing for starch. And you also need iodine, and you also need to take a record book, or you can have like a record data sheet to write down the observations in this experiment. So that being said, starch is an example of a polysaccharide, which is a complex sugar. So to test for polysaccharides or starch in this food sample, you have to add few drops of iodine to each of these food samples, like this. Adding like 3 or more drops to these food samples. So for the positive control group, as you can see, this is starch solution, and when I added iodine, few drops of iodine, you can see it's changing from brown to like bluish black, or let me just say deep blue. So the solution is turning to deep blue. For this, which is cassava flakes, as you can see, it's turning black, and that shows that there is a polysaccharide called starch in that food sample. Also in this food sample, this is yam. Starch is present because it turned from brown to black. Also, this is bread, turned from brown to black. Also, this is rice, same thing, from brown to black. And this is macaroni, also turned from brown to black. So this color changes means that starch is present in all these food samples. So in the positive control group, starch is present in all of them, and that's my expected result for each and every one of those food samples. So for the negative control, let's add few drops of iodine and see if there will be a color change, which will result to a false positive. Let's see. So in milk solution, we see that there's no color change. That's because there's lactose in this milk solution, and lactose is a disaccharide, so we shouldn't expect any color change because this experiment is testing for a polysaccharide, which is starch. In sugar, we see that there's no color change, and that's because sucrose is present in this solution, and sucrose is also a disaccharide, so that's why there's no color change. And finally, this is water. As usual, this is not a sugar, so we shouldn't expect any change. That's why it's under the negative control group. So observations in this experiment, as you can see, this is the data sheet for the experiment, and most especially all the food samples in the positive group turned black or bluish black, and there's no change in the negative control group. And so the negative control group has increased the confidence and the validity of this experiment, making this experiment valid. So, question for this experiment: why or what can be used as control groups? And basically what this question means is that what can you use for control groups in this experiment? So basically you can use any form of monosaccharide or disaccharide for this like negative control group, just to make sure that your experiment is valid. That means you can use glucose, you can use lactose, galactose, any form of monosaccharide or disaccharide. For example, there is lactose in this solution, which is milk solution. Here there is sucrose, which is a sugar solution, and ordinary water or distilled water, and those can be used as your controls or negative control group. And that's the end of this experiment. Thank you for watching this video. Bye bye.